Good morning. I would like to welcome everyone to today's MD Anderson Cancer Center Enjoy Science Summer Seminar. This is a series of lectures given by MD Anderson faculty members and organized by the Office of the Chief Scientific Officer, Dr. Giulio Draetta. I'm Alessandra Ferraioli and I have the pleasure of hosting this series with my colleague, Dr. George Kalin. The goal of the Enjoy Science initiative is to strengthen the connection between the scientific and medical community at MD Anderson and our colleagues, our patients, and our friends around the world during this time of limited in-person, at the bench, and in the clinic activities and interaction. This July 31st seminar is being recorded and will be made available on the Enjoy Science website later this week. The speaker of today is Dr. Moran Amit. He's an assistant professor in the Department of Head and Neck, Division of Surgery, the University of Texas MD Anderson Cancer Center in Houston, Texas. Dr. Amit received his MD from Tel Aviv University in Israel and completed a residency in head and neck surgery at Tel Aviv Suransky Medical Center and at the Rambam Medical Center in Haifa, Israel. Additionally, he obtained a PhD in cancer biology and immunology at the Technion Israel Institute of Technology. At MD Anderson, he has completed a clinical fellowship and a postdoctoral fellowship in head and neck surgical oncology and in head and neck cancer genomics. He is the recipient of many awards and honors for his clinical and research accomplishments and the lead author of numerous high-impact publications. Dr. Amit's research has covered numerous topics from cancer biology to cancer staging and cancer prevention. He has a particular interest in the neural regulation of cancer and has studied the mechanism used by the nervous system to promote tumor initiation, progression, and metastasis. Today, he will be discussing for us Cancer Strikes a Nerve, or how cancer acts our nervous system. Dr. Amit?
Hey, Alessandra. Thank you for the nice introduction. I actually give a lot of thought about what I'm going to speak about during this wonderful series of Enjoy Science established by you, the CSO, and Dr. Kalin. And I thought that the best idea would be just to walk you through like the journey through science that I've been through. I think that if it's gonna like affect anyone, you know, I earn my pay. I had no interest in science. I joined the military program for physicians back in the 90s. And I completed medical school, and then I joined the Army. I was a battalion doctor, participated, in things that happens in the Middle East, doing, general MD stuff. And then I moved to a Special Forces unit where I completed my military service there. And then I started my residency. And it was only then, after almost being 15 years in the business of medicine, when I started to pursue science and walking through the gates of science by Mao, one of my mentors, my clinical mentors. Let me just walk you through this journey. So I have nothing to disclose. So the first thing, so my training is as an ENT, as a head and neck surgeon. Was looking at patients with head and neck cancers. And the first thing we ask ourselves is, how are we doing? Like, are we doing something good? Should we change anything? And the way to look at it is to look at the trends of survival throughout the time. So this is one of the first papers that actually got published by our group, and we were looking at the outcomes of patients in the 1990s compared to those of the first decade of this current century. And we saw that actually we were doing something good. The survival rates went up for patients with oral malignancies, and the number of failing patients with distant metastasis went down. So the next question is, of course, what are we doing right? And there are many potential, factors that come into this equation. So, of course, the enhanced multidisciplinary care that was kind of integrated into the practice of oncology in the last, let's say, 30 years has become sort of the standard of care. In MD Anderson, of course, this is what we're practicing, but not every patient is privileged to come to MD Anderson, right? So, many people in the world are not actually have access to that kind of service. And it's really important to go and advocate for this multidisciplinary care in terms of each patient is going to see a surgeon, med-onc, rad-onc, those things that are trivial to us, but they became a standard. And another standard thing that became available to the patient population is, standardization of the verbiage that we're using, NCCN guidelines, AJCC staging system, those things that weren't there, back in the 50s, 60s, 70s, but they're available now. They're making us physicians speaking in the same language and treating the patients in the same way. Other things are related to, of course, implementation of clinical pathways that allows us to detect, recurrences earlier, like survivorship program, like we have in head and neck surgery, and of course, improvement in rehab and technology. Of course, we can't, move forward without it. So we have better imaging techniques today, PET-CT, metabolic imaging, that kind of things. We have a newer technology for surgery. Those things that we couldn't do in the past, like looking at the back of our throat. We had to open someone's jaw like a book in order to do it. This is why we stopped doing it and we started irradiating those patients. Today, we're using robotic instrumentation. This is like angular optics and angular instrumentation. Just imagine you can see behind, the tongue, and you don't need to open the jaw anymore. And of course, our fellow plastic surgeons that allowed us, with their advanced reconstructive technique and free tissue transfer from different parts of the body to the head and neck region, allowed us to do a more extensive resections with safer oncological margins and better outcomes in terms of functionality, morbidity, and of course, survival. But last and not least at all is science. We have a better understanding of the disease biology. So what we're looking at now is just one example. We are all doing science. I guess all the audience here is practicing some sort of, scientific endeavor, looking at different types of disease. So you probably have bazillions, examples for what I'm going to speak about. But this is just one example of one thing that we looked at. And again, today I'm going to speak about so many different, diseases and so many organs and types of cancer, because I want it to be, more of like, brainstorming with you guys, or at least, giving you some ideas. So this is thyroid cancer. Thyroid cancer, thyroid gland that right here in the front of the neck. Some patients gets, thyroid cancer, and one of the tools that we have to fight it is radioactive iodine. Thyroid is one of the only organs, this is a generalization, of course, that takes iodine, and we take advantage of it as clinicians. We put radioactive isotope of the iodine that release beta particles, some sort of a radiation that kills the cancer cells in the thyroid. So, but unfortunately, there is a group of patients that does not take it, that their thyroid cancer cells are not actually taking this radioactive iodine. And the reason is that their symporter that's supposed to take it from the membrane inside the cell is not working, or it's not there. We don't really know. We didn't really know. So we looked at the mechanism, and we figured out that one of the mechanisms is actually related to ribosomal machinery called the GPIT complex that does not allow the protein to fold and glycosylate in the way that needs to be in order for him to get to the membrane. So those sodium iodide importers are not functional, and they're not getting to the membrane. So this is just one demonstration. And of course, today there are ways to redifferentiate the tumors and treat this specific mechanism. But with better instruments and everything, you would expect that we'll have a lower amount of patients, and we'll have less patients and less disease burden, but that's not actually the truth. We have the same amount of patients for many types of cancer, and in certain cases, like thyroid cancer, for example, the number of patients even went up. So now we have a huge bulk of patients. The population is growing bigger. The diagnostics are getting better. We're diagnosing more and more patients with cancers. So we need to find a way to better stratify the patient, because we have more instruments in our armamentarium to treat the patients. We have radiation. Think about, like, we had radiation, chemo, and surgery. That's what we had. Today we have immunotherapy. We have targeted therapy. We have so many other types of therapy, but you have to tailor the right treatment for the right patients. So the first step would be to stratify the risk. The picture completely not related to what I'm talking about. I just love this picture. This is like we're going to get to it. These are nerves in culture. But in order to stratify the risk, we got to use some sort of a staging system. I'm not inventing it. It's out there. It's called the TNM staging. It's AJCC's, TNM staging. But can we do better? Can we actually stratify the risk better? Because when I was a resident, I felt that the staging system doesn't do a good work, So the way we looked at the tumor staging was very primitive. It was just based on size. If it's 2 centimeters, it's 1. If it's 2 to 4, it's 2. But there are so many other factors that can be added to this equation to make me look at the patient in a more personalized way and to tailor the treatment specific to this patient. Unfortunately, when I was a resident in Israel, I had not many patients. So I couldn't really make a statement about, we should do that or we should do something else. So I thought that the best way to overcome this obstacle of having just, I had like 100 patients. I couldn't really make a statement with that, basically, not even like particularly not on staging. So it's to create a collaboration, and this is one take-home message that I want you guys to take, or anyone that listens, to take from this talk today. You won't even imagine how eager are people out there to collaborate. You reach out to people and they're like, yes, let's do it. And they're actually doing it. We're creating the largest, highly or detailed annotated database for oral cavity cancer around the globe. We started with 9 centers. We ended up at its peak, it was 15 centers, over 5,000 patients with detailed annotated pathology. It's like unheard of. So when you're looking at SEER or something along those lines, you have a lot of clinical data, but you don't have really detailed pathology and outcome, which kind of updates periodically. So we'll look at the data of those patients, and first we'll look at the T stage. And when we looked at the T stage, again, the TNM stands for the tumor, node, and metastasis. The T is actually the primary tumor, how big it is, what other feature it has. So we knew that something is missing. For example, in melanoma, you have the invasion and you have the ulceration, but we don't have it in oral cavity because of cancer of head and neck. So we tried to incorporate, and we ran multiple models on it. And we figured out that actually if you incorporate one feature called the depth of invasion, that actually improves the performance of the staging system in a way that it's really stratify the risk of the patients in the right way. And if you're just using the old-fashioned, system, those T1, T2 kind of merge into each other, like you can see here, and T3, T4 have some sort of better separation, but for those patients with the early stage, it's not working. So we incorporated it, and actually we proposed it, and that became the standard. That's like the AJCC that was released 2 years ago, and adopted this system, and this is what we're using today in clinic. So we moved on to the nodal staging, and when we looked at the nodal staging, don't worry, I'm going to get to science in a couple minutes, hardcore science. But when we looked at the nodal staging, we figured out that even patients that did not have metastasis in pathology, but they did have suspicious for metastasis before the surgery, during the imaging or physical exam, for example, they actually were in higher risk group. You can see that their outcome were worse, even though they were PN0, meaning they had no nodal metastasis in the pathology after the surgery. They had higher risk and they had worse outcome. There are many, it's counterintuitive, but when you think about it for more than 1 minute, you figure out why. One is because we are a surgeon, we're not sampling good enough, so we probably kind of missed one of the lymph nodes that had metastasis, or as a pathologist, not all pathology suits are working like in Anderson, of course. So they're just cutting the lymph node in the middle, looking at it, oh, no mets, cool, PN0. But sometimes in order to detect this micrometastasis, you have to do serial sectioning or immunohistochemistry, and that's not being done routinely in many other places. So this is another way you can miss metastasis or micrometastasis. And we actually came with a lot of different, recommendation about elective node dissection and the number of lymph nodes and lymph node ratio that kind of factors those things in. It's a ratio between the positive nodes and the total number of nodes that gives us, it's kind of like controls for those variables of pathology, missteps, and the surgical sampling problems that I've mentioned. So we moved on, we asked ourselves, how can we do better in a little bit more orphan disease in head and neck? And we immediately looked at adenoid cystic carcinoma, which is a salivary gland tumor, a spit gland tumor, and it's very rare. I remember sitting with the database myself, I had like 15 patients, 1-5, that's it. So again, we used the same technique of reaching out to people that published on it, and boom, all of a sudden we ended up with like over 500 patients, with fully annotated clinical data and pathological data from around the world. We had 11 centers again. Unfortunately, the only continent that didn't participate in any of those consortia was Africa. I don't know why, but we didn't get any response from them, even though we reached out. So if anyone is here from Africa, please contact me after the talk. Would love to cooperate. And we looked at how those people fail, like how those patients fail, and we looked at the role of elective neck dissection. Meaning if a patient comes with no lymph node metastasis, just a primary tumor, should you do a neck dissection and take all those lymph nodes out or not? And we didn't know it, like until then. So we figured out that actually not all subsets in the head and neck are the same. So the major spit glands behave differently from the minor one in the mouth, and those in the mouth behave differently from those in the nose. So we defined all those, and we actually kind of set, the standard for, how to treat this disease in many ways. We looked at surgical margins, and we decided to look at one cool feature that this disease has, and this is neural invasion. This is where we start talking about nerves. So what's neural invasion? Neural invasion is the fourth route of metastasis. We all know about distant metastasis, hematogenous spread to distant organ. We all know about local invasion. We all know about lymphatic invasion, lymph node metastasis, or lymph node spread. But neural invasion is when the cancer cells actually take advantage of the nerves in the tumor microenvironment and uses them as a highway to spread distantly. So it can go to the spine, to the brain, to other organs through nerves, right? We see that, but we don't really understand what we're looking at or how it works. So we see a lot in head and neck. We see it in colorectal cancer, but the actual, I don't want to say pathognomonic, but the workhorse in the field are pancreas and prostate. Those are highly or densely innervated tumors. And we know that once the tumor hit the nerve, the patient is going to do bad. Overall survival is going to go down, recurrence is going to go up, morbidity is going to go up. Those nerves are affected. So this might be translated into pain. It might be translated into some functional morbidity. And this is a bad feature. It's so bad that in head and neck, for example, when we have perineural invasion, we give those patients radiation. That means that you cut all the tumor out, you put it under a microscope, no tumor in the patient, supposedly. But if in the specimen we see perineural invasion, we irradiate the patient in many instances. So it's that bad. Here are a couple of examples. This is a patient with sarcoma, and you can see that the lesion is here. This is an MRI scan, and this is coronal sectioning. You can see how the tumor actually goes all the way to the brain. From behind the mouth, the masticatory space, this is the space behind the mouth, it just goes to the brain. And this is another example of the nerve, the facial nerve. The nerve moves the face. How you can see how thickened it is here, but it's thickened because it's filled with cancer cells. And the nicest example, I think, here is actually a patient with melanoma of the skin, right here in front of the cheek, below the eye. You can see how thickened the nerve here. Just compare it to the other side. You don't have to be a radiologist expert. Just look, here it's so thick and white, and here there is nothing. This nerve here is actually what made this lesion unoperable. This is uncurable, basically. You can't chase the nerve all the way to the brain. This region is too critical. You've got like critical vessels that you cannot really access it. You can access it, but the patient won't do well. So With that in mind, we started looking at neural invasion in adenoid cystic carcinoma, and we knew that neural invasion doesn't come in, it comes in different flavors, in many different, types of neural invasion. So we know that in the upper panel, you can see perineural invasion when those cancer cells actually entrapping the nerve. They're circling they're encircling the nerve, but they're not really invading into the nerve like you can see in the bottom panel here. You can see like those tumor islands that are actually invading inside the nerve. This is called intraneural invasion in the bottom, and at the top is perineural invasion. And pathologists are not looking at it separately. They're saying like perineural invasion period, But when we looked at the outcome of the patients, we saw that those patients that actually had INI, intraneural invasion, did worse significantly for adenoid cystic carcinoma in every survival measure that we looked at. And so with that in mind, we thought, okay, so we're looking at intraneural invasion, perineural invasion, those are different processes, different outcomes, or kind of like the continuum of different outcome, but on the same spectrum. And we started looking at the mechanism. What causes that? Because apparently this tropism, those tumors that goes to the nerves, very much like flowers goes to the sun, it's not happening randomly. It's something directing it. So we proposed a couple of mechanisms, and we had a bunch of hypotheses, and we published it in Nature Reviews Cancer, and I like this figure. It was inspired by our article. You can see the tree is like the sprouting, nerve, and then you have the nest, which are like the cancer islands within it, and you can see the birds are going back and forth like the cancer cells along the neurons. And one thing that we was really important for us to state is this is not just cancer cells and nerves. No, there are many other players that support this game, and they are part of this game. So you have nerve supporting cell like Schwann cells here in the right lower corner, and you have immune cell, of course. You have immune cell all over, so why want to be in the nerves. So you have macrophages. And we actually kind of dissected all the mechanisms that were known. There are not many, you see all those molecules and receptors, but they're all focusing around, nerve growth factors and their associated receptors, basically. And we're going to touch it in a second. So my PhD thesis was on how the immune system actually mediates this process. And what you can see here in this nice video is how macrophages, endoneural macrophages, the GFP positive cells, are interacting with the pancreatic cancer cells in the bright field along an axon. You can't see the axon because it's very thin and it's not in the right focal plane. But I think that this short video kind of like demonstrates this interplay, this dance between these three factors, or three players within this process called the perineural invasion. And of course, we figured out, that those macrophages loaded with GDNF, glial-derived neurotrophic factor, which is part of the NGF family, are actually interacting with the cancer cells and make them more prone to invade nerves and crawl along them. And we developed it to look at the Schwann cells as well, together with our collaborator from Memorial Sloan Kettering, Rich Wong. He's a chair of head and neck surgery there, focused on Schwann cells. But we basically figured out that it's not just cancer cells and nerves, and it's not random. We dissected the mechanism. But the major question is that it? Are we talking just about, nerves serving as a highway for cancer to disseminate, or there's something beyond it, or nerves are actually actively playing a role in cancer progression and tumor progression and metastasis and other processes that we're all studying in our lab. The image that you see here in the background, this is a sensory neuron from a mouse labeled with the TRPV1, which is a sensory neuron marker. I thought it's a cool picture, so I just put it here. But yeah, it was cultured in our lab. Those neurons are fun to play with, but they're very spoiled. It's really tricky to grow those. So we know that the answer to this question is probably no. No. We know that nerves are actually playing an active role in cancer. We know that nerves are not just conduits, are not just fibers that something crawls along. And we know it from epidemiological data, and we know from population-based studies that were done out there, some of them here in Anderson, like this one, that showed that patients in breast cancer that were treated with beta blockers, one of the branches of the sympathetic nervous system, actually did better, here the yellow line, compared to those that were not treated with beta blockers in breast cancer. And we saw the same thing in melanoma, in skin cancer, and in prostate. So we know that actually if we modulate, if we manipulate the nervous system in a way that it won't function or it won't even be present in the tumor, probably or theoretically we hypothesize that those tumors will not progress or progress to a lower extent. So with that in mind, we looked at the data in head and neck, and we found these striking results. We found that when you have more nerves, as simple as that, more nerves equals worse outcome. Those patients that had higher neural density, or in a simple language, they had more nerves in the tumor, they did worse, they died more. And those that had less nerves in the tumor microenvironment, they did well, they just survived more. So one would have think that, okay, if this is so striking, why aren't we looking at it? So I just did some sort of exploratory search in PubMed. There are tens of thousands of papers of angiogenesis, starting in the 70s with Judah Folkman. And there are already clinical trials looking at anti-angiogenesis agents. Many of those are actively recruiting. I just looked at how many papers are out there on axonogenesis and nerve growth to cancer, and that was like 20-something. And when I looked at how many papers are actually looking at the mechanism, that was a single-digit number, even less than 5, 3 publications. And I wasn't even thinking to look at clinical trial on those, anti-axonogenic, factors trials, because they're not there, because we don't know what the mechanism is. Well, one hypothesis that we put back in the 2016 review paper was that this thing actually happens before the malignant transformation. And here's the thing that I never published or discussed. So when you're looking at the common denominator of those tumors that have perineural invasion, you see that many of them are just exocrine gland tumors, prostate, pancreas, salivary gland. And what's common to this tumor, they all have premalignant lesions. They all have, in pancreas you have pannins, or in prostate you have pins. So we kind of speculated that this thing happens before the epithelial cells actually transform into cancer cells, when they're still atypical, but not to an extent that we can call them cancer. And this is the preneoplastic lesions here that you can see. And you can see how the nerves, this is a diagram that we proposed in that paper, and you can see how the nerves started growing towards the preneoplastic lesion. And of course, when it's already, after the malignant transformation, after we have cancer, this is like a jungle. This is like a madhouse. You have so many players there, and of course you have the nerves. And here in the right lower corner, you can see how those cancer cells in red actually invade the nerve and the other factors participating. So in order to study that, we use the workhorse for this system, which is the DRG model. What is the DRG model? DRG model is basically when you take cancers, we take neurons, you put them together in the octagon and let them play. Just kidding, not the octagon, but in a dish. So you take the DRG from the spine of the mouse, you put it in Matrigel in the middle of a dish, you let it grow for 2 or 3 days to start sprouting, and then you put the cancer cells around it, or you can put it at the same time. And then you look at the interaction between them, and you can manipulate many, many factors in the media, in the cells, whatever. So here's a short video kind of demonstrating. In the bottom you have this cancer cell colony, at the top you have the axons. Coming from the DRG. This was taken in our lab when I was a PhD student. And what you can see here is see how nicely the cancer cells are detaching from the colony and start crawling along this axon filament towards the ganglia. Very directed, very specific, very orchestrated. It's not a random process. if you take those cancer cells, put them in a plate with nothing, they'll just push the margin forward, they'll grow, like a scratch test, basically. You've all probably done it before. You see it. And this is a nice picture that was taken in my lab, and it's on the cover of Cancer Research, but it looks like a bridge between the DRG right here in the bottom and the cancer cell colony. But see, you know, it grows on a filament. The filament serves as a foundation, as a highway, and it's not just random pushing margin. So when we try to apply it to head and neck cancer, this is when we found our striking, you know, result. So we put a lot of cancer cells, different cell lines, you know, with the DRGs, and we noticed that we're getting two types of neurons. We're getting those that look sad, like here, you know, all the filaments are kind of not growing, kind of like growing inside, and those that are sprouting like crazy, they're on fire. And the difference when we looked at the different cell lines that we put there, because the media was the same, was the P53 status. There were other mutations, but if you look at the common denominator was the P53. So we said, like, okay, let's try to replicate that in vivo. Let's try to inject those cells into mice and see what happens. So we did that, and when we injected the P53 wild type and the P53 deficient, we saw, and in green you can see the neural marker S100. There are better markers, but that's what we used initially. It's like the first picture that we took. We saw way more nerves in the tumor microenvironment compared to here, and you can see that the tumors are approximately the same size. So the next question, of course, is, does it happen in the pre-malignant phase? And of course, orthotopic is not the right model to test it, right? So we used a genetic model. And when you use a genetic model, the 4NQ-induced tumor here, you can see we used like two animals, like two types of animals, wild type and P53 deficient, and we gave them the 4NQ, the carcinogen, and we waited. After 20 weeks, you start looking at, you know, pre-malignant. After 30 weeks, you start seeing like the malignant, you know, invasive squame. And we actually found that it happens in the pre-malignant lesion phase right here, and only in the P53 deficient mouse. So with that in mind, we started contemplating, how does that happen? So if we're talking about pre-malignant, it probably happens before we have direct interaction between cancer cells or epithelial cells back then and nerves. So this is a paracrine effect. If we're talking about paracrine effect, the way I think about paracrine effect is a secreted thing. And secreted things come in two ways. Either just secreted or wrapped into boxes like Amazon boxes and shipped, which these boxes, we call them extracellular vesicles or exosomes. So in order to figure out which one is responsible, we separated those compartments. We used ultracentrifugation, established protocols to look at the exosomes separately and the rest, all the soluble things that are excreted to the juice, you know, what you have above your cells in the plate. And we found there was only the exosomal compartment of the P53 deficient cells that actually causes sprouting of those DRGs and not the secreted factor. And when we looked, of course, at the wild type, we didn't see anything, right? Those are our exosomes that are taken by Kenneth Dunner from the EM facility, a great guy and a great friend, and a great scientist. How nice our images are. So this is a nice demonstration of those exosomes, how they're taken by the nerve. It's 3D reconstruction using the confocal with Jared Burks at the North Campus microscopy facility. So we looked at the exosomes labeled with DIL inside of nerves labeled with beta-3 tubulin that were cultured together. So, okay, we figured out those are exosomes. So what's in the exosome can cause that. So one major component of the exosome content is actually RNA. This is one I call George. And the RNA, we wanted first to dissect, you know, the difference between the RNA content and P53 mutated or null. So we sequenced the RNA within them, and we got, our top hits, for the differentially expressed microRNA within the exosomes. And when I saw the 34, I was so excited. I knew that 34 is a master regulator of differentiation, of stemness. And I knew that upstream to it, it's regulated by P53. So it gave me like the direct links to everything. But we still tested many of the other differentially expressed microRNA. So the way to do it is it's very laborious. You have to test them one by one. But let me show you what happened when we tested it. So we know that those 53 wild type have a lot of microRNA. I'll go back. They have a lot of those microRNAs, these ones. So what you need to do is take the exosome, put it on neuron, and you, like I showed you, they're not supposed to grow because this comes from wild type. But in order to pinpoint which one is responsible, you have to block them one by one and see which one will cause these nerves to sprout. And what we did is we blocked 34 using integument and see what happened. These neurons in the plate started sprouting like crazy. So we figured out that the lock to this mechanism is probably 34, and we have to get rid of it. But reading a lot about microRNAs, I thought that and knowing a lot about neurons, I thought that just a single microRNA cannot really make this effect. It has to be an orchestrated either by other factors or other microRNAs. So we looked at the literature. We came up with a list of microRNAs that are involved in this process of axonogenesis or nerve invasion. And they actually existed based on the sequencing that we did in the inside those exosomes. And we came with this, again, after testing them one by one, with this combination of microRNAs that causes those nerves to sprout. So we're talking about miR-21 and TREE24 that are stimulatory. They're stimulating the growth of those neurons, but the removal of miR-34 to allow it to happen. If I'll use an analogy, if you have a door, it's not important how hard you're going to shake the handle. It's not going to open if the door is locked. So the handle are those two miRs, but the lock is 34. Once you remove the miR-34, those two microRNAs start working on the nerve and causing it to sprout. So when I take the three of them and put them together and transfect it into the neurons, nothing happens. It's only when I remove the 34 when you start seeing this sprouting, this neural growth of in vitro. So with that in mind, we asked ourselves, okay, so but what types of nerves we're looking at? Because, we have many different types. We have sympathetic, parasympathetic, we have sensory, we have motor. So we see a lot of neuron. This is cool, but what types of nerves and how can we manipulate that to make cancer history, right? So we started staining. We labeled them for different, markers of sympathetic, parasympathetic using P53 wild type and P53 mutated samples from, human patients that we had in our lab. And we figured out that the difference in the nerve phenotype was mostly in the sympathetic. So TH stands for tyrosine hydroxylase. This is one of the enzymes that are involved in the catacomb in biosynthesis. And we can see here that in the TP53 mutated or P53 deficient, you have high expression of it, which you don't have in wild type. You don't see, difference in the markers of parasympathetic nerves when you compare wild type to null, but you do see in sympathetic. So we went back to the animals and we looked at their ganglia. So we injected the tumors, we harvested the neurons in the skull base right here. They sit like if you open the cranium of the animal and you lift the brain right below. And we looked at it and you can see when with no tumor that how it looks like, no TH at all. With wild type you get like few cells, scattered cells here. When you look at P53 deficient, look how many cells. And we confirmed it with flow, the number of cells, to show that they're actually increasing with in the presence of P53 deficient tumors. But the most interesting thing would be to look whether those tumors are different from the endogenous tumors whether those neurons are different than the endogenous neurons in the animal. So we ran mRNA-seq to compare neurons derived from a wild-type animal and neurons derived from a p53 deficient tumors and compare those. And as you can see here, they're completely different. And when we put together using pathway analysis, when we put together what pathways are involved, we figured out that those genes differentially expressed are related to neurotransmission, proliferation and exon sprouting and branching and neurogenesis and neuritogenesis and stemness, of course. Because here's the thing, so neurons are the most mature cells in the human body. In order to redifferentiate, they have to go through a phase of dedifferentiation, not all the way to pluripotency, but to some sort of like a midway, and then go back to their new differentiated state. So, and here you can see this is like an IPA analysis of the genes that came up in the previous slide. So you can see that the pain signaling, because those are sensory nerves. Those are the nerves that actually innervates our face when we touch our face, or our tongue when we touch our tongue. You can see that the pain signaling actually goes down, but not completely. The pain transmission system is not shut down completely, but it seems to have like an acquisition of a new feature, which is the catecholamine biosynthesis and sympathetic signaling. So we ask ourselves, okay, so that's cool. We have sympathetic nerves in the tumor. Let's kill them and then see what's going to happen. And we did that. We used the 6-OHDA, which is a dopamine agonist that actually kills specifically sympathetic neurons. Guess what happened? Nothing. Nothing happened because we did that before we implanted the tumor. So you're killing the pre-existing sympathetic neurons, and then you inject the tumor, and then you're not expecting new nerves to come, right? But that's exactly what's happening. Because if you remember when I told you, those nerves are growing to the tumor as part of the process of tumorigenesis. And but the question is what the origin of these nerves? And I showed you already that those are redifferentiated sensory neurons. So we said, okay, let's cut the sensory nerves. We did that and see what happened. So when you cut the sensory nerves, surprisingly, those nerves are related to sensation, right? They're not supposed to do anything. They are afferent. They're not efferent. So they're not even signaling to the tumor. So why is it happening? So we cut those, and we repeated this experiment so many times because it was so mind-blowing for us. So we cut those nerves, and boom, the tumors are shrinking and are not even growing. This is like a non-growing tumor from our standpoint. So again, we asked ourselves, where did they come from? We knew that they are coming from sensory nerves. So one nice thing about the oral cavity is that it doesn't have a lot of sympathetic innervation, if any. The only sympathetic innervation comes from or belongs to the perivascular space. It's confined to the perivascular space. Every nerve that you see outside of this space is aberrant, is anatomically abnormal. So what we did, we implanted those tumors, which are p53 deficient, that's supposed to induce growth of nerves to the tumor, and we cut the nerves before. And see what happens. When you cut the nerve, you just don't get this new cancer-related sympathetic neurons. They're just not growing. And I remind you, we didn't manipulate the sympathetic nervous system. We cut the sensory. So you cut the sensory and you don't get growth of sympathetic. This is like, wow. This is sham surgery. We just opened the skin and closed it. This is just controlled and nothing happens. That means that everything happens as planned. So you get like those new neurons. And if you remember, I told you about, they have to go through a level of dedifferentiation. So we looked at stemness factors, and we found that those actually p53 null expressing stemness features, which are sympathetic reprogramming markers like the OCT4, KLF4. I guess many of you heard about those. And ASCL1, or in humans, it's MESH1. Which is fascinating factor because it's directly related to sympathetic system development in embryos. So that was a proof that we are looking at dedifferentiation and redifferentiation, or transdifferentiation in one word. So of course, we're a clinician, so we wanted to see how we take advantage of it, and we tried to treat it. We saw that we have a lot of sympathetic nerves in the tumor, so we want to block it, so we use the beta blockers. So we induce those tumors, and we use beta blockers with Corvedlo, which is non-selective beta blocker, and we evidence a significant reduction in the number of, in the size of those tumors in two different cell lines. Both has either PCI13 PBAB is 53 deficient, and HN31 is 53 mutated. And when we looked at the cohort of patients we had, we labeled it for TH. We found that those patients that had a lot of nerves positive for TH actually did worse. And you can see it in red. They had like a worse survival and more recurrences just based on the count of the number of TH positive neural filaments in the tumor microenvironment. So kind of to summarize everything, and I love this cartoon. It was made by my brother-in-law, Yal. He's a tattoo artist, and you're going to enjoy his art a little bit later. So when we lose P53, we have a very unique composition of microRNA within those extracellular vesicles, also known as exosomes. And this is being released to the tumor microenvironment and taken by the nerve. And once it's taken by the nerve, those microRNAs get to the cell body of the neuron and start running a transcriptional program that actually reprogram or redifferentiated those neurons to something else than what they used to be. So those are the involved, transcription factors. And once it's acquiring those adrenergic or sympathetic features, what happens, it starts feeding back the tumor with adrenergic signaling that causes the tumor to grow, metastasize, and eventually kill the patient. I think this is the last scientific slide. I just wanted to share, some of my experience with you guys, and ladies, of course. And I just wanted to bring those things that I believe in. I think that the one thing that you have to do in order to make an innovative research program or start innovative research is to, first you have to challenge the paradigms that you're working with. Don't take everything, as for granted, challenge it. We challenge the staging system. We challenge our understanding about nerves. Once you figure it out, that not all the brains are sitting in one room, in your room. It's probably you have to collaborate, build a team, either to obtain samples or to obtain inputs or to do some things that you're not able to do. And always go back and assess your data. And you have to be rigor about it, and you have to be meticulous about how your data looks like. With that, I'm going to end. Let's see. We'll have enough room for questions. I'd like to thank my mentors, Dr. Myers, the chairman of my department, Dr. Kalin, you all know, Dr. Hanna is the director of the skull-based program with which I worked a lot on skull-based tumors, skull-based melanoma, and SNUC, rare tumors. I want to thank my friends and colleagues. Hideaki Takahashi was a postdoc with me when I was in Dr. Myers' lab. He's now back in Japan. He is like my soulmate. Mihnea Dragomir, back in Romania now. Great guy, funny guy, knows everything about everything. Simone Fossi, fortunately, still here in Houston. We're working together, a great friend and a great scientist. Our funders, NIH, NCI, and TCR on the Moonshot program. Myers lab members, North Campus Microscopy Facility, EM Facility, my collaborators, and the list is very long, on the ICOR Consortia and the NICC carcinoma, my collaborators in Houston Methodist that helped us with making those artificial exosomes, and of course the head and neck biorepository personnel. With that, I think I'm going to adjourn. Thank you all for being here with us today and listening. I'll take some questions. I hope you enjoy this cartoon. made by, again, Eyal, the two artists. Going to be in the background for 6 minutes, so you have 6 minutes for questions. Thank you all.
Thank you, Moran. This is a great presentation, and now this is why MD Anderson it's such a nice place to work, because there are people like you and many others who combine wonderful clinic with wonderful science. And now let's start with a question. So first, are there any control clinical studies showing prevention of neural metastasis with beta blockers? They make a lot of sense for many cancers that metastasize to brain.
As of now, there are no studies like that. We are proposing a study like that in non-melanoma skin cancer. We are actually in the process of writing the protocol these days, but there are no studies that are manipulating nerves. The only study that I'm aware of, and it was done in prostate, not with beta blockers, but with Botox, with botulinum toxin in prostate, it was stopped because they weren't able to demonstrate any efficacy.
Okay. Now another question. I would like to know why p53 loss was chosen first for studying neural invasion. It is because in oral cancer p53 is mutated a lot, or because Myers Lab studied p53 a lot? Also, why microRNA analysis is chosen over protein secretion analysis and so on? So basically, he want to know the detail, how your mind was working.
Yeah. This is a great question, and we actually dealt with it. So with regard to the first question, as I said, we used many different cell lines, and we had this observation that I remember this lab meeting actually very vividly. So we had two types of DRGs, those that sprouted and those that were very quiescent. Nothing happened. They looked just a black dot. And when we looked at the different cell lines that we used in these different dishes, the common denominator of those that were quiescent was that they were p53 wild type, while those that were sprouting like fire, those were p53 either mutated or no. So this is with regard to question number one. What was question two again? Oh, the microRNA. MicroRNA, we focused on those. We actually tested everything. So we looked at the proteome of the extracellular vesicles, but when we compared wild type to p53 null, we didn't see any difference. We ran in Rebiotic ELISA assay for nerve growth factors. We couldn't see any difference. So they're there, but they're not different between wild type and null. So we didn't focus on those. We preferred to focus on the microRNA because of this reason.
Another question. What triggers neurogenesis during invasiveness in the cancer?
microRNAs.
I let you to say. It's.
Well, that's us. I was just kidding. I guess it's like a symphony. There are many players there. We found out about the microRNAs and their ability to reprogram neurons and to induce neurogenesis. Probably we're speaking of other factors as well. And connected to the previous question, probably some NGF, GDNF, maybe artemin. Those are the major players in the field. Artemin was described by Gustavo Ayala back in 2008 in a clinical cancer research paper. The other two were mentioned by us when I was looking at perineural invasion. The other paper that looks at the neurogenesis don't actually look at how it happens. They just, describe its occurrence and describe the cell of origin. And I refer to a Nature Communication paper from 2018 on oropharyngeal cancer and Magnon's paper from Nature that was published a couple of months before ours was published in Nature that just showed that where those cells are coming from. And actually in this paper, their findings were similar but different. They were similar because they showed that those are neural stem cells, which is kind of what we found in a way-ish, but theirs were suggesting that they're actually traveling from the brain all the way to the prostate. which sounds like wow to me, that a normal cell, like a normal organ, will metastasize through the bloodstream, cross the blood-brain barrier, and go to the prostate.
Great. Now the same person connected the question. So what triggers neurogenesis? And come with a second question. Are perineural macrophages doing so to trigger neurogenesis through perineural macrophages?
I haven't showed it, but my short answer would be absolutely. I haven't tested it, but they secrete, I've showed that they secrete GDNF, which is a strong neurogenic cue. And definitely they play a role. Even though I didn't test it, I think that it has to be part of the game. Yeah.
Another question. In the clinical trials that you propose, it would be good to include some safe drugs to suppress stem-like phenotype. Any thoughts?
This is a great idea. I've never thought about it. We were thinking about looking at beta blockers first because those are safe and you know, the toxicity profile has been out there for like 50 years. Everyone's using it. But this is a great thing. Like I'll need to read about it because I'm not familiar with those agents.
Now, another question that come from me. So you presented all the data on the research part on oral cancers. How general do you think the mechanism that you show it's in human cancers in different histotypes?
I think that this phenomenon, we see a lot of nerves in prostate. We see a lot of nerves in pancreas. We see a lot of nerves even in breast and gastric cancer. I think that those highly innervated or densely innervated tumors definitely share this mechanism, but it will have to be, you know, shown by others, I guess.
Good. Another question. Please continue.
No, I just want to say, I think, again, it's an hypothesis that this mechanism will be valid for other types of cancer, but I haven't demonstrated it, so it's hard to comment on it.
Okay, so you have enough time to continue to work on this mechanism. So another question. Are there studies showing neural invasion leads finally to brain metastasis?
Sorry, go ahead.
Yeah, so he was saying also, sorry, you might have answer, I miss it. So if there are studies showing if neural invasion leads finally to brain metastasis.
Right, so there are many publications, mostly from the field of radiology, because they love those images that I showed you, like the MRI and the CAT scan. As a clinician, I can tell that many patients come with a small lesion here, and then we track how backward the tumor goes along the nerves. And in many cases, unfortunately, those patients become inoperable because the tumor goes all the way to the brain. So the short answer is yes. Those are not, you know, case control or something like that. Usually those are, you know, kind of case reports and presentation by radiologists that are showing those cases because they're very interesting, and we'll have to educate, you know, the professionals out there to look for those things. Because if you don't look for it, you won't notice it. And the only thing that might give you a hint about it is a patient that comes and say like, oh, I feel a little bit numb here, or they can't feel their forehead because the tumor that started here, like over their eyebrow, goes along the nerves and actually paralyzing it. So they become like numb and they don't feel the pain and they don't feel anything. And it crawls all the way along the skull base to the brain. So yes, there are, you know, papers there. If the person that asked the question is interested, they should look for the term perineural spread. This is the term for a spread to distant organs like spine and brain.
Another question. Have you compared the neuronal function from tumor edge versus in the core versus normal tissue in head and neck cancers versus non-head and neck?
We're doing it now these days. We're using cohort. This is a project I'm doing with Kate Hutchison, Leon Gross, and a fellow Gibran. We are looking at oropharyngeal cancer patients, and we are profiling the neural networks in those tumors. And the cool thing about this study is that we have functional data. We have a patient-reported outcome and functional data on swallowing and pain, and we're going to correlate how those nerves looks in different regions of the tumor, like the person that asked the question just discussed, and the functional outcome of the patient. So this is a great question, and we're just doing it these days.
Thank you. So with this, Moran, thank you very much. It was a wonderful presentation, very interesting data, both clinical and scientifically. Also, I have to say, instead of you, I have to say for you, guys, Moran it's opening his own lab, so certainly he has position. So if you have good ideas, with his good ideas, I think will be a great combination. And with this, I come back to all of you, the ones who are here each Friday with us. Without you, such webinars make no sense. So thank you for being here with us. Thank you again, Moran, for your talk, and keep in mind, all of you, science has this wonderful characteristic. Science unites people from different colors, different religions, different places of the world, because what is best and most beautiful is making discoveries to treat cancer patient and patient with any type of disease. With this, next time we invite you Thursdays for the month of July. We will have moved on Thursdays. And next time, Thursday 13, it's a wonderful presentation by Jeff Moldren. So we are looking to all of you to be with us in 2 weeks from now. Thank you again, and thank you, Moran, for your wonderful presentation. Good luck.
Thank you. Thanks for having me, and good luck to you all. It's been great to be with you.
Thank you. Very inspiring. Thank you.